Hello and welcome everyone. Welcome to Adult Bee Disease presentation today. My name is Anne-Marie Fauvel and I'm the Tech Transfer Team Coordinator for the Bee Informed Partnership. This is kind of a part two of a series of webinars that we did at the Bee Informed Partnership. This one is really about honeybee adult diseases. We have another one on brood diseases, which covers American foul brood, European foul brood, chalk brood, and parasitic mite syndrome. And so we will not be addressing those diseases today. But if you are interested in those diseases, please you are welcome to visit our website at beeinformed.org and visit the tab that says BIP Talks. And on there, you will see a series of webinar recordings. And brooding over brood, I believe, the brood disease one is right there, and you are welcome to view it as well. So the Bee Informed Partnership, for those of you that don't know, has the great mission of improving colony health. We try to do this by bridging the gap between science and industry, and we do this even more so by supporting beekeepers in their practices. So the Tech Transfer Team program, one of our many programs, has field specialists in 5 hubs in the US. So one in Texas, California, working with mainly California queen breeders, but also other beekeepers in the Pacific Northwest, covering Oregon, Washington, Idaho, and Montana. In the Minnesota region, they go through Minnesota as well as North Dakota. We also have a field specialist in the Great Lakes region covering Wisconsin, Michigan, as well as New York. And those field specialists have a unique perspective on the bee industry. They have their hands, ears, and eyes in tens of thousands of colonies every year. So they have accumulated a wealth of information, and some of that information we will share with you today on how to look for adult bee diseases, diagnose them, and perhaps even prevent some. So in today's agenda, I think that we need to start with colony collapse disorder. My colleague, Natalie Steinhauer, who is the science coordinator for BIP, will be talking about CCD. There is quite a bit of misconception. The media has used this term as a catch-all term. And so I'm hoping that Natalie will disband some misconception and misinformation about the term itself and the phenomena. I will then take up the Honeybee Viruses section, where we're going to talk about transmission routes, some of the viruses, and also ways to prevent it. And then Kate Houston, our very brand new field specialist in Texas, will talk about Nozema ceranae. And Dan Wines, our Michigan field specialist, will talk a little bit about Tracheal Mites. So without further ado, here is Natalie Steinhauer with CCD.
So we wanted to start off by really addressing the elephant in the room and rectifying a lot of misunderstandings surrounding the issue of CCD. Now, if you search CCD on the web, you cannot not find press or published works that alarmingly describes the doom that awaits us if all of the honeybees were to disappear. Now we all agree that there are concerns surrounding honeybee health and just, environmental quality and loss of biodiversity in general, but it is important to really separate the issues and define the issues so that we know what we are talking about. So I would like to start with a poll to kind of like judge where you stand on the following statement. Which of the following statement is correct? So we'll give you a couple of seconds to address which of those statements you think is correct. And then we will address that and then tell you which one is correct and then why the others are wrong. So we obviously are all in agreement that honeybee health is not ideal. There are issues, there are concerns, and we at BIP, we are documenting the turnover rates of colonies every year. We have run the loss and management survey since 2007. A lot of you have mentioned that you have answered the survey this year, and we thank you. So we know that every year about 4 out of 10 colonies in the US will die off and need to be replaced. So this shows a mortality rate, right, which can be matched by something like a natality rate, right? So for colonies, that means that creating new colonies by having swarms or making splits can be the counterpart of losing colonies. So it is not the same as a population loss. It is really a turnover. A lot of people are worried that honeybee populations are in decline, And to look at that question, we need to estimate the total number of colonies in the country. And this is something that the USDA does every year in their honey reports. So looking at those numbers, we see that there was a reduction since the early 1960s, in which the US had about 5.5 million colonies. And more recently, we have been hovering around 2.5 million colonies in the United States. Now, we can talk about this graph in more details, and particularly about how the incidence of CCD and the arrival of Varroa mites in the country, they have barely impacted it. And by comparison, the biggest drop in population estimate was actually due to a methodological change in how the survey was conducted. But in short, I have added here the arrow showing since when BIP has conducted his loss and management survey. And so since BIP survey started documenting 40% annual turnover, the population, the total number of colonies in the country has remained stable despite high mortality, despite high turnaround. So that means that beekeepers can maintain their operation size, they can recover from their losses, and the population as a whole is relatively stable. Of course, that comes at a cost, and we don't want to underappreciate the efforts it takes beekeeper to do that. So I just wanted to give you in comparison, to give you context during the same time period. So this is in orange, we show exactly the same data as before. The scale is different because now I'm also showing you the European Union and the world numbers. So this is the estimate of the world number of honeybee colonies. And you can see that honeybees are actually increasing in the population worldwide. So there might be some regional declines, but overall honeybees are neither in decline nor at risk of extinction. Again, this is despite high turnover on the colonies, because beekeepers can make new colonies and maintain or increase their operation size. So this is the first, I guess, main point from this presentation, is that whenever you talk about colony loss mortality rate, it is different than honeybee population decline, and it is different from CCD. So those are three different phenomena that have different drivers and different consequences. So what about CCD? Well, okay, CCD was an outbreak. It was a cluster in time and space of high mortalities with really, really weird features. The first cases were declared in the fall of 2006, and then it became more widespread in the spring of 2007. And the beekeepers were reporting large and unusual die-offs. Now, what was unusual was the way those colonies died. They described that thriving populous colonies would be found mostly empty in a few weeks, with no dead bees that were found in or around the hive. Now, that drew a lot of attention because this is why we started hearing about the vanishing bees, right? Those bees just suddenly disappeared. But actually, this particular sign, no dead bees found, was actually not completely unheard of. It is actually called altruistic suicide, and the bees that are sick are known to do this. So they will walk away, they will die outside of the colony. And this has been observed in multiple infectious cases. For example, high Varroa infestation can trigger altruistic suicide of adult bees, and other infection can cause that behavior as well. So it is believed to be an evolved response to minimize infection inside the superorganism that is the honeybees. So that on its own was not unheard of. But what was really particular, and so when the researchers described CCD, they actually coined several key signs that all have to be present in order to qualify as CCD. So all the colonies that found dying or dead from CCD, they shared this common set of clinical signs. The first one was the rapid loss of adult bees from the colony, as indicated by large areas of brood that remained unattended. So you can see in the figure above, we basically had excess brood population relative to the adult bee population that wasn't enough to cover all the brood. And this was really rapid. As I said, in a couple of weeks time, you have this rapid loss of adult bees. The second, of course, was the absence of dead bees in the colony apiary, as I said, probably due to altruistic behavior. And then the other sign was the fact that there was sufficient stores of honey and pollen in the affected colonies, meaning that they didn't lack in food, they didn't starve. And again, another sign was the apparent delay in kleptoparasitism in abandoned hives. So that means robbing or invasion by small hive beetles or wax moths. So you probably all know, if you keep bees, that if you have unattended frames inside a colony that doesn't have bees, they will get attacked by those opportunistic pests, and that can arrive very, very quickly. So that was a weird symptoms in which that kleptoparasitism, those opportunistic pests, would either not come or come really later. So despite the fact that basically the food stores were undefended. And then finally, a little later, they added to the definition that the levels of Nozema and Varroa had to be insufficiently high to explain the collapse. So they ruled out Nozema and Varroa from CCD cases. They ruled them out as causes, and they said, in order to qualify as CCD, you have to have low infestation of those two pests. So you can see how each of those signs can actually be expressed in multiple bee health conditions, but all together they describe CCD. So having one or two is not enough. You really need to have all of them to qualify to the strict definition of CCD. And so then you can see now how this term can be misappropriated by a lot of the press out there, and what the issue is. So we talked about that the first case description was done in 2007 in the Cox-Foster publication, which I've shown a print screen here. And then researchers actually looked at historical reports, and they found that such losses actually had been documented in the US at least 18 times in the last 110 years. And they were reported under different names like May disease, disappearing disease, and never really clearly defined. So when it was defined in 2007, that's when they made the case definition, listed all of the signs, and made the actual case paper. So to date, it actually has only been documented with verified case only in the US and in Switzerland. And still today, no definitive cause of CCD has been identified. The original study implicated a virus, which was IAPV, in combination with Nozema as a potential causal agent. But then the association was not confirmed by a second, larger study that had more samples, and that actually followed over 200 different variables to try to find a source that would explain the cases. And still no single factor was found in all cases. They even found that pesticides that are often suggested as a putative cause for CCD were higher in healthy colonies than in colonies affected by CCD. So still a lot of unknowns. They did notice that colonies dying from the condition were clustered together, so hinting that the condition seemed communicable, or the result of some common exposure rather than some large-scale change in the environment. And they also found that the bees collected from collapsing colonies had higher pathogen loads and were more often co-infected by multiple pathogens rather compared to healthy colonies. So it did suggest that maybe the immune system of the bees were compromised, letting then the bees succumb from opportunistic second infection. So a little like AIDS in humans. So what now? Most experts, most apiary inspectors in our country, and our own field specialists at the Bee Informed Partnership agree that we have not encountered cases that meet the strict case definition, at least in the last 5 years, if not 10 years. Our oldest BIP field specialist team was established in 2010 in California, and none of us have ever seen an official case of CCD. So definitely CCD is not that prominent anymore. It really acted like an epidemic. It had a peak of case in 2006, 2007, and then stabilized back under detectable levels. We are still unsure what the cause is, but we know that it is not a major cause of honeybee mortality anymore. However, it did act as a highlighting and attracting the attention of the press and the public to long-term issues on honeybee health. So it really acted like a whistleblower. But it is, unfortunately, has been misused in the press to mean more than it actually is. So if you really follow the case definition, CCD is not around anymore. It was a flared epidemic. It is in line with the virulent pathogen hypothesis, though the original causal agent has not been identified. It is possible that it might flare back up from time to time. And if it does, we hope that then the new molecular analysis will allow us to identify a causal agent this time around. But currently, it is not something that we see. Now, unfortunately, if you use the definition as used in the press, which is mostly misappropriated as any problem touching honeybees, any lost colony, then yes, I guess CCD continues because we do see high loss rates in many places. The problem of using the term in that way is that it kind of hides the fact that with a proper management, many colony loss can be averted. So it is important to disambiguate issues because there are some issues that can be averted, and hiding them under catch-all terms kind of hide that fact. Now, when we ask beekeepers, however, a lot of beekeepers still report CCD as one of the causes of colony loss in their operation. I'm showing you here over 8 years of survey data in which we ask the question, what do you perceive as the major cause of colony loss in your operation? And we noticed that about 29% of large-scale beekeepers have reported it. It is actually the fourth most important cause of colony loss reported by large-scale beekeeper after Varroa, queen failure, and pesticides. In comparison, only just 7% of small-scale beekeepers report it. But so we can see that it is a perception problem and probably is a definition problem, and that's why people are still reporting it. So we know that colonies can be lost by, due to a lot of different factors, and a lot of things can go wrong in a colony, but it is important to try to identify the reasons a colony die, because that's how you can try to make sure that it doesn't happen in the future. That's how you can understand the issues and remedy it in the future. So over the years, our field specialists have been called to sites where people have reported CCD, but they always found that there were other stressors, other causes to those issues. So remember the list of signs associated with the definition of CCD, and remember that all must be true to qualify. So if you do have dead bees in a bunch at the entrance of the colony, then it is not CCD. If you have high mite infestation, it is not CCD. If you find an empty colony, but it doesn't have any brood left, it is not CCD. So finding a dead colony in your yard, we know it's a harrowing experience, and figuring out what killed it is tricky. It does take experience, but we hope that it's an exercise worth practicing, and we hope that we can be helpful. For example, in this webinar, where we can try to give you tips on how to do this identification.
Next, we will discuss honeybee viruses. I would like you all to note that the original recording of this adult bee disease presentation included this section presented by Rob Snyder, our California field specialist. But I had to re-record it due to microphone issues throughout the recording. So I apologize. I have tried to keep close to what he presented in the first place, but you may hear other field specialists later on in the presentation referring to what Rob said earlier, when in fact it was Anne-Marie, so that you might understand. So viruses are an emerging field of research in the bee industry, and much more research is desperately needed in this area, and so much remains unknown. Since the introduction of Varroa mites in 1987, viruses have become more and more virulent, and beekeepers must pay really close attention to their colony's health, Varroa mite treatment, as well as management. Some research has shown that once viral loads are high in colonies, it can take up to 2 years or more to rid the colony of high viral loads through diligent Varroa mite treatments, requeening, and supplying bees with good forage. The best way to keep viral loads at a minimum is to monitor for Varroa mites and treat the colonies when they are close to 3% threshold. Although many of us here at BIP, we believe that in the spring anyway, mite loads should be 1% or less. So here is a list of viruses. You must remember all of these by the end of the presentation, and you will be quizzed on it. No, I'm kidding, of course. I wouldn't even put this list up normally, but in previous presentations, participants have asked for a cheat sheet. They were interested in knowing what each of those viruses looked like, and they were distracted throughout the rest of the slides because they didn't know what those viruses were. So here are some of the viruses that we look at when we do viral analyses. We look at acute bee paralysis virus, chronic bee paralysis virus, deformed wing virus, Israeli acute paralysis virus, Kashmir bee virus, sackbrood virus, slow bee paralysis virus, and Varroa Destructor virus, which is also known to be the deformed wing virus variant B. There's some fighting in the research with the researchers about this deformed wing virus. I'll talk about it a little bit in the next few slides. So this is a very busy slide, I realize, but let's see if I can explain and simplify it a little bit. Here are, I wanted to review the different ways in which viruses can be transmitted. So mainly there are three main transmission routes: the horizontal transmission. So that's, generally from B to B through oral or fecal route, and direct contact. So this transmission is used by most viruses. So that whole list, you can basically put that in this particular type of transmission. Bees are highly in contact with one another, and most viruses can be transmitted this way. Another transmission route is the vertical transmission, and vertical transmission is from mother and or father, so from queen or drones to the daughter bees. We'll have a different slide to show you how that works too. And lastly, viruses can be transmitted through a third party, or what we call a vector, namely Varroa in the case of honeybees. So we'll talk about some examples of viruses that are vectored by Varroa in the next few slides. So viruses can be transmitted again through these different routes. Traditionally, viruses were transmitted horizontally. It's the most popular way, I guess, from B to B, again, or either oral or fecal route, or simply by contact, like in the illustration here. We demonstrate this kind of transmission. So adult bees, nurse bees in particular, come in contact with larvae at a young age through feeding, sometimes cannibalism when they need to take care of a dead or diseased bee, and also when they clean cells, for examples. Adult bees also come in frequent contact with other adult bees through grooming and sharing of food, as well as just simply contact surfaces. So example of viruses that are using this type of transmission, as I mentioned before, are basically all the viruses on the list. It's a pretty popular way. No worries, you don't need to remember those viruses again. Just know that most viruses are transmitted this way. Viruses could also be transmitted vertically, mentioned earlier, by either mother or father to their progeny. And in this illustration, we can see that either a queen or a drone can transmit viruses via the egg or via the drone sperm, for example. This transmission is fairly straightforward, and examples of viruses that use this type of transmission include acute bee paralysis virus, or ABPV, deformed wing virus, or VDV1, which is Varroa Destructor virus, also known as deformed wing virus B variant. And then lastly, the transmission route that is the most important factor that determines the dynamic of pathogens, in this case, Varroa. Viruses can be transmitted in direct ways through a third party, also known as a vector. And this has been really important in the last few years. So the recent spread of Varroa Destructor greatly modified the epidemiology of several viruses, which switched from low loads to mostly orally transmitted pathogens to rapidly spreading epidemics driven by the direct transmission and or indirect effects of Varroa on individual and colony bee health. So shortly after this invasion of Varroa, viruses such as acute bee paralysis virus, deformed wing virus, and slow bee paralysis virus were shown to be successfully transmitted by the mite. And some of these viruses rapidly evolved towards a vector-borne lifestyle, meaning that they chose this particular transmission as their favorite way of doing this, and they're the fastest way for them to kind of conquer the honeybees. And it increased, of course, the rate of prevalence as well as the virulence in the US mite population, and of course, in turn, in the honeybee population. So examples of Varroa mediated transmission or vectored by Varroa mites include acute bee paralysis virus, Kashmir bee virus, Israeli acute paralysis virus, deformed wing virus, VDV1, Varroa Destructor, also known as the deformed-wing virus variant B, and then the slow bee paralysis virus. Dr. Coulinet followed 20 colonies in seven different locations in the US, and she found that the Varroa loads throughout the course of 3 seasons was closely associated with the deformed virus A load. So in this here, you can see the Varroa load by months. So first month and the 6th month. So the first month, I believe, is May, and the 6th month is October. So it represents spring going into summer, going into fall. And you can see that at the end in the fall, you have the two, most of them are all above threshold. However, you can see that throughout the season in the blue line here, you had much lower Varroa loads, and in fact, lower than the threshold for most of the season, compared to the average, which is on the yellow or orange line here, where you have higher Varroa loads throughout the season and going above threshold fairly early in the summertime. And if you superpose that with what she found out for loads of the deformed-wing virus A, you can see that that line in yellow that was earlier in the season higher in Varroa mites showed a significantly greater load of the deformed-wing virus A. So specifically, the number of times in which colonies were above threshold was associated with higher viral loads in the fall. I think that this is very telling, and I think that it's important to remember that. So remember to keep your mites low early in the season so that you keep your viral loads low in the fall. So here's your virus test list again. On the right-hand side, you can see, find the visual representation of the viral loads that we put together in our viral processing reports. So unfortunately, one of the pieces of information that's missing in the research is that we currently don't have a viral load threshold the way we have a threshold for Varroa. We don't have a viral load threshold for each of the viruses yet. And so instead of, telling you you're too high, you're too low, or giving you a threshold to show you where you're at, the one thing that we can do is we can show the results by comparing it to all the other results that we have in our database. So we can tell you in which percentile your results fall. So are you in the 25th percentile? So you're lower than the average beekeepers that we have analyzed results for? Are you in the 90th percentiles, meaning that you are higher than 90% of the beekeepers we have results for? So basically, in other words, we can't tell you if a certain level is too high, but we can tell you how it compares to the loads that we have in our database. So let's look at some visual signs of viruses. The visual signs are really few and far between. Very few viruses display visual signs, but a few do. For example, most of you have probably seen the misshapen, deformed, shrunken-like wings of honeybees, and that's a sure sign of deformed-wing virus. What you may not know is that if bees have these kinds of wings, they are pretty much sure to have deformed-wing virus, but the opposite is not true. If they don't have deformed wings, it does not mean that they don't have the virus. So the deformed wings may be a sign of the virus, but it is not always present. And this is one of the reasons why some of the researchers prefer to call this virus Varroa Destructor virus, because they feel like it's not really appropriate to call a virus by a sign that may or may not be there. And so we do know that it is vectored by Varroa, and that is the reason why they like to call it Varroa Destructor virus. In any case, this is a sure way to diagnose that you have the virus if you see this in your colony, but not the other way around. Here are other photos of these deformed wings. Bees with deformed wings cannot perform their functions in the colony, and therefore they have a much shortened life expectancy. So you can see those wings. It's kind of a sad affair. Another prominent visual sign is the dark coloring of a developing queen in its queen cell. These two images here show a clear sign of what we called black queen cell virus. It's pretty nicely describing the virus, isn't it? But again, the virus can be present in a colony and in the absence of visual signs. And in fact, we did a quick case study, Rob did in California, and he basically looked at the viral loads for a bunch of colonies in which he had found these black queen cells, and in some colonies where he did not find any black queen cells. And in the end, when the results came in, the viral loads came back fairly similar between both groups. We also see black queen cell virus appear in the majority of the viral samples that we process in the lab, but we don't really understand its full effect on colonies just yet. Another visual signs of virus is the shiny thorax and body of bees, which can be a sign of chronic bee paralysis virus. This visual sign can lead to misdiagnosis, however, because it can also be a sign of other condition or even simply the result of old age. So when I encounter this sign, when I do an inspection, I always note it and I look for other indication in the colony that something may be wrong before I make a diagnosis based on solely that sign. And lastly, the final visual signs of viruses is this underdeveloped pupil head. And it's usually uncapped and it presents itself like on the photos here on the slide. And this is often a sign of sac brood virus. But here again, one needs to be really careful about making a hasty diagnosis because it can also be an early sign of chalk brood. So the best way to know is to actually pull the larvae out of the cell for further examination, further investigation. And so it looks something like this. You can see here, I don't know if you can see my pointer, but you can see at the bottom of the cell, when you pull it with a pair of tweezers like this, you can see at the bottom of the cell, you see it better in the middle photo here at the bottom. There's kind of a fluid filled sac at the bottom. You can see it here as well. And then if you don't see that fluid sac, you may want to search for other larvae and pull them out and see if you can find it there. Or conversely, look for other signs of chalk brood because oftentimes it is that underdeveloped pupil head at the top can be a sign of early chalk brood. So while in the field specialists are often called to take bee samples to send for viral analysis. And what they do is they usually collect and ship live bees, or they can collect 50 milliliter tubes of bees that they freeze right away and send to the lab. Once they get to the lab, they will use approximately 50 bees, make a nice little mixture, use solutions to clean and amplify the viral RNA, and then quantify the number of viral RNA copies. This uses what they call the qPCR technology, and it neatly spits out the results into a report for beekeepers, which is really kind of cool. So finally, what you all want to know is how to prevent and treat viruses, right? Well, as you are probably well aware by now, after the events of this year, there are no treatments against viruses. Unfortunately, the answer is not very sexy here, and you're probably very tired of hearing it. But the only thing and the most important thing that beekeepers can do in order to prevent viruses is to monitor and treat for Varroa. At the risk of repeating myself, the only thing beekeepers can do to prevent virus transmission, and the best way they can do this, that can be achieved, is by controlling Varroa loads in their colonies. I really wish we had more tools to combat viruses, both human viruses these days and honeybee viruses. But alas, all we can do is slow down the transmission. So please, please, please monitor and treat your colonies for Varroa. Okay, enough about viruses. Let's now talk about Nozema with Cade.
Hello everyone, my name is Cade Houston. I'm the Texas Tech Team Field Specialist, and I'm going to talk a little bit about Nozema today. So before we get into talking about how Nozema is in honeybees, we need to know what it is on a biological level. So Nozema is a microsporidia. Microsporidia are eukaryotic unicellular organisms allied with fungi, but they're actually placed in a sister kingdom simply because they're hard to place in the nomenclature with fungi. So they're intracellular obligate parasites as well. So intracellular obligate parasites affect the gut of the host. So obligate parasites cannot complete their life cycle without a host, as is the case with Nozema and honeybees. So the picture in the middle is a comparison of two different bee guts that have been pulled out. The top one you can see is clearly lighter in color than the one on the bottom, and that light color suggests that it is infected. And the picture on the right is a microscope look at Nozema, and that long yarn-like thing you see there is actually the polar tube, the polar filament that they use to infect the midgut cell. And we'll talk more about that in the next slide. Okay, so now we're going to get into the life cycle a little bit. So Nozema spores enter the midgut through contact with other bees via trophallaxis, which is mouth-to-mouth contact or presence of feces in the hive environment. So once the Nozema cell reaches the folds of the midgut, that long polar filament, it'll shoot that into an epithelial cell, into a midgut cell. And it releases the reproductive organism there, and those cells begin to replicate, and they replicate until that cell is just so full it breaks. And those Nozema cells are released, and they go in and infect other bees inside the hive. Okay, so the two types of Nozema that affect honeybees are Nozema apis and Nozema cerani. Nozema apis was discovered on Apis mellifera, the European honeybee. It made its way into the U.S. by the early 1900s and was believed to be the only type of Nozema that affected honeybees for a long time. That was until Nozema cerani came along. And the first report was in Europe in 2006 of Nozema cerani. But it was believed to have been in the U.S. since the 1990s. It was first discovered on the Asian honeybee, and but now it affects other Apis species and also non-Apis species. So pictured here are on the left are Nozema apis and Nozema cerani. So as you can tell, there's no way to distinguish the two species under a microscope. So molecular analysis is the only way to distinguish the two, and we no longer speciate between the two because Nozema apis has not been seen in years. Therefore, from now on, in this presentation, when we describe Nozema, we will be talking about Nozema cerani. Okay, so dysentery was often associated with Nozema apis, but not with Nozema cerani. Therefore, nowadays, if you see dysentery, it could just be a sign of spring cleansing flights if the bees have been stuck inside the hive for a long period of time due to cold weather or other environmental factors. Also, different types of forages will cause dysentery. So, if you see this dysentery, it's most likely a sign of another environmental stressor. Okay, here's some more potential signs of Nozema. Visual detection is very unreliable, but Some of these signs include K-wing, disjointed abdomens, absence of stinging reflex, or bees crawling on the ground. Just do note that these are potential signs of other conditions as well. As Rob pointed out earlier, and Dan will point out later, other conditions such as bee paralysis, virus, starvation, and pesticide poisoning, bees do exhibit those signs with those other conditions. Okay, so here, this graph here is the APIS longitudinal data in regards to Nozema. So as you can see, in 2018, 65% of apiaries tested positive, or approximately 65% of apiaries tested positive for Nozema. This was assumed Nozema cerani. So the infection intensity or average Nozema load, as you can see there in the graph, is measured in millions of spores per bee. And you can see that it fluctuates throughout the season. There is a slight increase in spring, which may be due to the bees are coming out of cold weather. They haven't had the chance to really get out and do their cleansing flights. So the levels might be higher. And also, Nozema is a old bee disease. So your older age bees inside the hive are likely to have higher spore counts. So you may have, your queen's just getting started, really just getting laying, and your brood production is really just starting back up. So the population in that hive is going to be a lot of old bees. So you may have that may be a reason that we're seeing higher counts coming out of winter. Okay, so here, this is the graph that Natalie used earlier, and as you can see, on average, about 15% of large-scale beekeepers identify Nozema as a source of colony loss, and only 4% of small-scale beekeepers identify it as a cause of colony loss. And just another note, the fraction of beekeepers reporting Nozema as a cause of colony loss has decreased over the years. Nozema ranks 9 out of 16 in the order of most important causes of colony loss in large-scale operations, and 10 out of 16 for small-scale. All right, so now we'll go into some Nozema diagnostic techniques. So microscopic examination is the most reliable and most used to account for presence and quantity of Nozema spores. So in our lab, we don't use dissection. So we take a sample of bees, crush them with a rolling pin, which makes sort of the insides of the bees are crushed out, so it makes sort of a bee juice. And a sample is taken from this, and it's analyzed under the microscope to quantify the amount of spores that are present. And you can do this at home. Just get a certain number of bees, crush them up, and take a sample and look at it under a microscope if you have one available. So those are microscopic, just to tie back, microscopic examination is really the most reliable and most used way to diagnose Nozema. All right, so now we'll go into some prevention methods. So to prevent Nozema, beekeepers want to have good brood production, and you can't have good brood production without proper nutrition. So you want to make sure your bees have enough honey and pollen. You want to ask yourself that question: do my bees have enough honey and enough pollen stores to successfully rear brood? You want to make sure that they have everything they need to make that hive grow, to make that population grow, and to really amp that queen up and get her to lay more. And so, I mean, you can feed through feeding pollen supplements, sugar water, corn syrup. You just need to make sure that those bees have enough feed, and if not, the management practice would be to feed them. All right. The next thing is you want to have a young, prolific queen, and that equals good brood production. So just that's another thing with the good brood production. You want to have a having her in that hive will give you that large population of younger bees. As we described earlier, the Nozema is an old bee disease. You want a high population of those young bees uh, to uh, to prevent the Nozema. Um, and actually, in fact, high Nozema spore counts oftentimes do go back to queen issues in many cases. So a good management practice would be to monitor the age of your queens and the production rates as well. Make sure those queens are producing to the best to the best of their ability, and yeah, your hives will be that much better for it in Nozema prevention. Okay, so next thing is choose good apiary sites. So you want to choose good sites with good forage, which goes back to good nutrition, which in turn goes back to the good brood production. So sites with good forage, proper ventilation, good water drainage, and good site accessibility. So like we said earlier, like we just said, the good nutrition goes back to good brood production. Um, Nozema is closely related to fungi. So they like those moist, humid conditions. So good water drainage and proper ventilation is completely necessary to prevent Nozema. Um, and good site accessibility, that's more on a management for the beekeeper scale, because if you don't have good site accessibility, you can't get in there to feed, to medicate, to requeen if needed, and all these things play together in Nozema prevention and are connected. So it's good to have these three. Okay, so now we'll go into some ways to treat Nozema. So there are two ways. You can eliminate the spores uh, through treating your equipment, or you can treat using fumagillin, which is on the market. And we'll get into that on the next slide. So here uh, you can use thermal decontamination, which you want to get your equipment up to 120 degrees and hold it at that temperature for 24 hours. This will kill the Nozema spores. So, and then irradiation, it has been known to kill AFB spores, which in turn will most likely kill the Nozema spores. And then fumigation with acetic acid, ethylene oxide, and phostoxin. Okay, so there's only one treatment available to treat Nozema on honeybees in the United States, and that is up until well, fumagillin. It is the only approved treatment for Nozema in honeybees. It was removed from the market last year, but it's now available again as Fumidil B. And although it was removed from the market, then brought back, beekeepers never reported a huge colony loss due to Nozema in that year where fumagillin was not available. Okay, so on this graph, you can see the spore loads in millions on the y-axis and the years on the x-axis. So what stands out here is seasonal load swings occur regardless of treatment, regardless whether the beekeepers treat it or not. And there's no significant difference between treated and non-treated. The graphs are fairly similar. I mean, you can see the peaks and the declines, they're very similar. So in conclusion, with Nozema, it's all a matter of preference for the beekeeper. Like, do you treat or do you not treat? That's the real question with Nozema. Do you want to treat or not? And I think our next spokesman is Dan Wines.
Yeah. Good afternoon, good morning. Dan Wines here. I am the tech team field specialist based in Michigan, kind of covering the Great Lakes region. And I'm going to talk about tracheal mites. Here we have this this photo just to kind of see this. So this kind of stripy appearing tube is an extracted trachea from an adult honeybee, and these kind of sort of roughly oval shape, football shape kind of darker patches within that trachea, those are the actual mites. So we get a sense we're dealing with something pretty small here. It's unlike the Varroa mite. It's not something we're going to pick up with our own eyes. It does require a microscope, and we'll talk about that in a minute. Just to give a bit of brief background on this parasite, it was first discovered on the Isle of Wight, which is just an island off the Great Britain. 1921, they'd been having through the 1900s, 19-teens, they'd been having a lot of high losses. It kind of really spawned a lot of advances in bee science and investigation, trying to determine what these high losses were caused by. And in this investigation, they were dissecting bees, and they found these tracheal mites within honeybees. And so that was where they were first described. It was kind of later determined this wasn't the cause of these high losses, but it did lead to the discovery of this parasite. Interestingly, shortly after they were discovered, the United States passed the Honeybee Act of 1922, which banned the import of honeybees from infected countries. So they kind of an early on the biosecurity measure there. It did spread through Europe pretty quickly after it was found, or it was found in Europe shortly after it was found in the United Kingdom. Despite kind of these bee shipment regulations in the United States, it did cross the Atlantic, was found in Brazil in the 70s, and then in Mexico in 1980. First confirmed finding in the United States was in Texas in 1984 in the summertime, and by that fall it had been found in a number of different states. So it was pretty common with the ways bees get moved around the country for pollination, for honey production, for wintering locations, for shipping of nukes, packages, queens, things like this. Once something kind of gets here, it's everywhere pretty quickly. So this next slide kind of shows us what happened. So this is from the USDA Bee Diagnostic Lab, and what it shows, these blue dots represent essentially each year the percentage of colonies that were the percentage of samples that were infested with these tracheomites. So we see this black line here, 1984, when they first arrived in the United States. It's very low, it's pretty rare, but immediately the next year we're up in the 35% range, and that number stays pretty steady in the 35-40% of samples analyzed had tracheomites for about a decade. We go from kind of the mid 80s into the mid 90s. Now the second line I put on this graph, this vertical red line, is 1987. This is when the Varroa mites turned up in the United States, and similar story, they were kind of detected in one place, and then within a matter of a couple of years they were pretty well everywhere. As we went through the 90s, it became increasingly imperative that beekeepers treated for Varroa mites to keep their colonies alive and healthy. And what the thought, why we have this, as we kind of see this, as we look at the blue dots, this kind of essentially a trend line down to zero, is that in being forced to treat for the Varroa mites, beekeepers were actually killing the tracheomites as well. Kind of, if you're familiar with the concept of like bycatch, you're targeting one thing, but also in this case happens to be a positive outcome is we're killing this tracheal mite as well when we're using treatments to kill Varroa mites. So we see this essentially trend line to zero in the within the last decade. They're rare enough that the National Honeybee Survey, APHIS, removed them from the monitoring efforts in 2011 because in 2009 and 2010 they were not detected in any sample within that survey. And currently the USDA will still process samples to look for them, but they report about a 1% positive finding of tracheomites on an annual basis. So not something that's particularly common these days, but it was, say, a couple decades back, it was a major part of beekeeping. So just a little bit on this, it's a parasite. On our next slide here, I have kind of a close-up picture of it. And it does live exclusively, as far as we know, exclusively on Apis mellifera, or European or Western honeybees. It's an internal parasite. It lives exclusively in the trachea of adult bees, except for there's a brief period where they transfer bee to bee, but they're living in the bee in the tracheal system, which is part of the respiratory system, and in that trachea system, they essentially are chewing a hole through that trachea to access hemolymph, which is what they feed on. This next slide kind of outlines the life cycle. So it basically begins when a female mite detects. She's able to detect air, kind of the outward breath of the bee, if you will, from a spiracle, which is kind of a hole at the end of a trachea in the bee's thorax. So it's kind of the end of the line in the respiratory system. And so our female mite is able to detect that, and she's able to detect that she's able to pick out a young bee, typically between like 1 and 4 days old, that she's going to enter the respiratory system of. Once she's in there and lodged in the trachea, she lays eggs. Typically, it's about half a dozen within the first few, 3 or 4 days. She'll lay both female and male mites. Typically, the female mites outnumber the male mites 3 or 4 to 1. The males do mature a little quicker, 11 to 12 days. Females are 14 or 15. But once they're both male and female mature, they do mate, and that kind of process takes about 3 weeks total to get them eggs laid, matured, mated. They develop through from egg to larva, nymph, and then finally adults. They mate, and those mated females exit the bee. This bee now started as a young bee a few days old when it was infested. Now it's somewhere in the range of approaching a month old, so it's kind of an older, maybe it's a forager now. But this mature mite exits the bee. She will kind of make her way to the end of one of the hairs on that bee's thorax, and she will look for a new young bee to reinfest and start that cycle over. And because of this kind of old bee to young bee transmission, it was kind of determined that most of this bee to bee spread within the colony occurs at night, or when there's inclement weather, rainy days and things like that, or cold temperatures when the bees aren't flying. So, and if we think about this, this makes sense, because this is a time when we have that high concentration of bees. Kind of everybody's at home if nobody's flying, and we have that contact between the old bees and the young bees all being kind of tight together in there. So it's easier for that mite to be riding on a hair of an older bee and detect and then jump onto a younger bee. So it's kind of the basic cycle of the way this mite kind of progresses in its life cycle and transmission from bee to bee. As far as its impacts on the adult bees, it's one of those, it's tricky because there is no reliable visual indicator. Sometimes you hear reported K-wing, which came up earlier. This is kind of where the bees have 2 pairs of wings, and you'll see just in their resting position, like this bee on the right, its 2 sets of wings are splayed apart. Normally they have between their wings, there's a little piece of anatomy called a frenulum, which kind of it hooks the hind wing and the forewing together, so they kind of appear as one set. But when these are splayed out like that, that can be a sign of tracheal mites. Another potential visual indicator is a distended or kind of bloated abdomen. Sometimes tracheal mites can lead to crawling bees at the entrance. But as we've kind of discussed in these other stressors of the colony, these are not reliable and specific to this particular, they're not specific to tracheal mites. There's a lot of things that can cause bees to present these visual symptoms. As far as colony level impacts, beekeepers report, there's kind of this thought of like it's the colony's failure to thrive. Everything is there that should be there for healthy growth. It's a time of year when you'd expect your colony to be, building and growing. There's good food in the environment. The weather's favorable. It's a time of year when you would expect a lot of growth. You don't have, Varroa mite issues or other things like that. And the bees are just kind of stuck and stalled, and they don't go forward. You see diminished brood area, the population's smaller than it should be. They don't grow. The honey yields are not going to be there, even if, if it's there in the environment for them, and they're just not able to go out and gather it and put on that surplus weight. And then particularly problematic when we get to the winter, we see a looser cluster. It's, so our winter bees need to cluster together, and they're in doing so, they're thermoregulating to essentially survive the winter in cold locations. And with this ability to thermoregulate being disrupted, it disrupts that clustering behavior and leads to an increased winter mortality, particularly in northern locations. When the bees, or when the tracheal mites first arrived in the States, it was really much more problematic for beekeepers that overwintered in the northern regions. They saw a big spike in losses due to this kind of disruption of clustering behavior. So again, a lot of things in colonies can kind of lead to these, a lot of stressors will lead to diminished brood area and, colonies not growing. So again, no real reliable visual diagnostics, but these are kind of some of the things that you may be looking for that may, put in your mind tracheal mites. What is required for detection or for diagnosis is dissection. Here we see this photo on the left is a slice of a bee's thorax, and these kind of two roughly vertical white tubes that kind of have a little bit of a V kind of kink in them. Those are the bee's trachea part, which make up the respiratory system there. And we can see on the right, these are kind of segments from the trachea that have been removed from that thorax. And the top photo on the right, those little kind of discolorations, the kind of yellow, darkish kind of lumps, those are actually the mites in there. So when the dissection is looking for mites, they're looking for discoloration, cloudiness, kind of scarring in the trachea. Normally they should be kind of clear and uniform in color with kind of no abnormalities. And then this picture at the bottom is just, again, another piece of trachea that's been removed with those two little, blips on the bottom right there outside the tube. Those are just to kind of give a sense. Those are tracheal mites. It gives you a sense it's fairly large relative to the trachea that it is infesting. So that's what's required to kind of get a confirming diagnosis. When I say, like, the USDA reports, 1% detection rate, they're doing this kind of dissection to look for the tracheal mites. As far as prevention, they do spread between colonies via drift. As I say, that bee to bee interaction is how they spread from individual bees, and that's how they're going to get from colony to colony. So just thinking about laying out your apiaries in a way to minimize drift. Also, not inciting any robbing events where you're getting a lot of interaction and transfer from bees between colonies. Those kind of just general good beekeeping practices for a lot of different reasons, but it can help there. There's also been, some genetic help through bee breeding, where they've kind of found that bees that practice grooming behavior have a better resistance to tracheal mites. And that makes sense when we think about this little brief window where the mite is outside the bee on that hair, looking to get from its old bee to its new host of the young bee. So bees that are grooming are going to break that transmission. And really, though, the main thing, as we kind of saw in that graph that I opened with, it is the Varroa control that has really brought the tracheal mites down to kind of a rarity these days. So good management practices with Varroa, being on top of it, using multiple methods, and being aggressive on controlling Varroa seems to be in that kind of bycatch. By doing that, you're very likely that you will kind of prevent tracheal mite issues as well. Having said that, there is one treatment approved for tracheal mites. As with anything, just like with our Varroa products, it is important, the label is the law. It's important that we understand how and when to apply these, both for bee health, but also for human health, for our health as beekeepers, but also not to contaminate honey or things like that. So the treatment that is labeled for tracheal mites is menthol crystals. They're applied in a kind of a mesh bag. They're available from bee suppliers. Just as we think about with our Varroa mite treatments, there are some that can be used with Supers-On and some that cannot be. This one for tracheal mites is something you cannot use with Supers-On, so it's going to be an early spring or late summer treatment. Typically the treatment is between 15 days and 6 weeks. Again, this is where you want to read the label and make sure you're applying it appropriately. Like we see with some of our Varroa treatments, there are some temperature considerations. So this one, the recommendation is that between daytime high temperatures of 60 and 80 degrees, the bag of menthol crystals should be placed on the top bars of the top brood chamber. There is also a note that above temperatures, if temperatures are anticipated to exceed 80 degrees, that instead this bag of menthol crystals should be placed at the bottom of the hive. So, and we do have one, if you get a positive diagnosis for tracheal mites, there is one product out there that is approved to treat them. So that's kind of the rundown of this pest. And, just to kind of summarize before we move to questions, what we've run through here. And say tracheal mites, they were an issue. I think they're an interesting kind of part of the beekeeping story of the last several decades, but they're not something we really need to be too concerned about today. I think it's good to be aware of them, but they're not a major stressor at this point. As Natalie talked about with the colony collapse disorder, it was a big deal, 14, 15 years ago. It's not something that we really are currently seeing. That we get a lot of reports, but as she said, a lot of this is kind of the misconception and co-option of this term and what it actually means, and misuse of this term in the media. So it's good to be aware of what it was, and it's not to say that we may not see these things come up again, but currently it's not something that's a major concern. Nozema is a little more of a middle ground, as Cade kind of talked about. There are some impacts on colony health, but there's a lot of unknowns. It's one that we as beekeepers and bee scientists kind of puzzle and scratch our head on. What the Nozema numbers mean, and should we be treating, and if we are treating, are we getting good value out of that treatment? Are we achieving our objective? So that's one. There's plenty of ongoing work to understand this Nozema. I say when we transferred or switched from kind of Nozema apis to Nozema ceranae, it was kind of caused this. We need to relearn what this parasite is and how it impacts our colonies and what we as beekeepers can do. And then the last one is, as Rob talked about, the viruses. These ones are what I see as a beekeeper and working with beekeepers and bee scientists. These viruses they're big problems. They are very much in the category of we're learning more every day about them. There's a lot of work going into understanding them because they are so problematic. It's really become a research priority of a lot of scientists because say we're starting to understand these mortality more, and it's not just the Varroa mites, it's the viruses that come along with them, and the long time that it can take a colony to kind of rebound once they've been seriously impacted from this. So
thank you, Dan. Thank you, Cade. Thanks, Natalie, for this great presentation. I hope all of you have enjoyed them. I need to stop by this slide real quick and thank our sponsors and collaborators, all the universities that we do research with, and all the sponsors that provide us with some funding and some projects that we can do in the field. So thank you to those. If you have any questions, please feel free to email me. My email is at the bottom here at fauvelam, FAUVELAM@umd.edu with any of your questions or your comments regarding this presentation. We also are a small nonprofit organization struggling like everybody else, and even more so in these COVID times. So if you have a few extra dollars and you found any value in our presentation today, please do not hesitate. We strongly encourage you to come and visit the beeinform.org website /donate and make a contribution to the Bee Informed Partnership and the work that we do. Thank you again. Thanks for watching, and we hope that you have enjoyed.